Yes, good morning to all. You can complete your task maybe later. It's about a 3 o'clock time is given, right? So you can keep it at aside. We may have our today's work. Yeah, put aside. So in this part, today I'm going to take you in a lab also that we are going to inoculate our liquid medium. Some growth culture will be given to you, or the plates may be given directly. You are going to use your nichrome wire loop to transfer a loopful culture to your liquid mediums. I think you have prepared at least one tube for yourself, right? It was more than that, but more than that are there. So you may go for at least 1 or 2 tubes can be inoculated. After the inoculations, you may keep for the incubations. So on a next, today is like a Thursday, right? So Friday you are coming tomorrow. So we may go for in a break or somewhere we need to arrange something that you are going to observe them. It is required to do. Observation is required. Otherwise, if you are going to observe after 1 week, then it is of no use. So tomorrow I do have one lecture or not? Okay. On a Friday, I think I don't have any lectures, right? Okay. So contact. It will take about 20 minutes, 10 to 15 minutes. See, you need to observe your tubes. We are going to keep one control. Okay, so at 12 o'clock we can go. એ જ કહું છું આજે લેબમાં જાશું ઢોસા બનાવો હોય તો આગલા દિવસે ખીરું બનાવીએ ને પછી બીજા દિવસે ઢોસા બને એટલે આજે આપણે ખીરું પેલા લોટ બાંધીને રાખેલો હતો આજે એમાં આપણે ખીરું નાખશું ખીરા માટે યસ not for the agar. It is for the liquid broth, nutrient broth you have prepared, right? Where you can do streaking. What I'm telling that we are going to inoculate only liquid broths. Just one first part we have done. Second part you are going to perform in the next lecture or next practical session. Okay, so I will brief on practical sessions about it. What we are going to do for a 10 or 15 minutes. Then we will move in a lab. Okay. So first part in this, what we were starting. You can put your attendance. So till now we have seen the 3 major techniques. These all 3 techniques are of a 3 different practicals for us. Okay. After completing it, we are going to prepare for the solid medium. On the same solid medium, on a next practical, you are going to do a first streaking. Okay. Then in the second part, into it, that if time will permit it, we will going to perform both the experiment together. That will be a streak and the spread, because it will not take time more. Because the streaking it will require about 5 minutes to 10 minutes to handling it. Same at a time, you can go for the second part as a spreading. So in the next lecture, we are going to prepare the solid medium. Okay, in the next practical, that plates, same plates you are going to inoculate, that is the streaking will be done, and you are going to for a spreading. 4 plate method, it will require another practical generally, because in a 4 plate, now we are going to learn about it. I think you have just completed with streak and spread plates. I hope there is no any doubt anybody. Okay. Yes, till now, just I was able to show it. Okay, so let me show that the documentary, how the spreading is done. Yesterday, we just left here. Same use person. Same as use person. I will tell you about the practical session during practical time. Now concentrate over here. It will be better for me and you. Okay.
The spread plate method is a technique to plate a liquid sample containing bacteria so that the bacteria are easy to count and isolate. So it is a very important technique for a microbiologist. Welcome to the channel Microchem's Experiments. Be
It is
with
very
us,
basic
be a
things,
laboratory
it's
expert.
going to say
To
about
perform
how
this
the
method
spreading
we
is
need a
done.
biological
Last
safety cabinet,
road is
incubator,
we are going to use.
micropipette, vortex mixer, micropipette tips, prepared fresh culture media plate, Bunsen burner, glass rod spreader and colony counter. So let's begin with the culture
It's
media
showing about the
preparation.
first, how many minimum requirements are.
Take weight of agar media and mix with distilled water following the label on the media container.
First, prepare
Sterile the
plates.
media according to the manufacturer's instructions. For sterilization, some media need to just boil at 100 degrees Celsius to dissolve the media content. Some need autoclaving at 121 degrees Celsius and 15 pounds for 15 minutes. Others need to boil in microwave oven for 2 minutes. After sterilization, pour the hot agar media in sterile Petri dishes and wait to cool and solidify.
So plates
Before
are
use,
ready.
incubate the
Till now.
plates for contamination checking.
Blank plates have been kept for the incubation.
After incubation,
Check
sort out the fresh
any
plates for
contamination.
using these in the test. Now we are going to inoculate our sample into previously prepared culture media plate by spread plate method. First of all, wet a glass spreader by spraying 70% ethanol or isopropyl alcohol.
Yes, these are the glass rods. It is like a L shaped. Okay, having the bend, 3 bend will be there. Okay, so it is a L shape. There we can use. Sometimes people are spraying the alcohol on the glass rod, but it is not advisable because when you're spraying, it is maybe near to the jar, then it may catch the fire. So it is generally not advisable, but it's depend upon the person to person and practice to practice from place to place. Okay, so what we can do, we can have some beaker. Within the beaker, we may take the alcohol, and over there we are going to dip our glass rod. And that glass rod, it is hot burn.
Alcohol and burn it by
For a few
rotating for
seconds.
sterilization.
The alcohol is going to burn out. You are not going to make it hot for a longer time. Otherwise, it will take a long time to get cooled down. Hey, it is made up of glass, but it should not be get.
After that,
Hotter
keep it
and
inside
hotter
the
a
biological
longer time.
safety cabinet and wait to cool. Label a fresh culture media plate.
So
Labeling
label your
should be
plates
done by media
at the
name,
bottom.
test name, method, date, etc. Bring everything needed for the spread plate technique.
So you can see the micropipettes. Okay, that micropipette minimum requirement for you people right now.
Take the sample 1 and vortex it to make a homogeneous mixture.
You can use the vertex, otherwise you can tap it also. Your tubes require to tap.
Shake and
I will
invert
going
the
to show you
sample
how to
several
tap.
times before
Water,
taking it for testing.
Tap.
Take 0.1 milliliters of the
Let I will
sample 1 by a
tell you.
micropipette and pour it drop-wise on the surface of the agar culture media plate.
So local culture, not but when spreading, we are going to take a certain amount of
Discard the tips from the micropipette. Start spreading the sample on the agar media surface and rotate the plate with finger periodically during mixing to ensure the proper spreading. Continue spreading until the sample portion is absorbed on the media surface.
It will take about 2 to 3 minutes at least for completing your task.
Stop spreading if you feel that the media surface is dried up. Now invert and incubate the inoculated plate in appropriate growth conditions, according to the media used and microorganisms tested for. After the incubation period, take out the plate and observe the result under the colony counter. You can see many isolated colonies of bacteria in our cultured plate. Now we will count each of the colony forming units to interpret the result.
Just to show
We got 36 colonies.
here how just basically this colony counter meter is been used. Here it is some chart is been prepared. Okay, that chart you can draw in your notes. That is like you are preparing the graph, right? So it is the number is there. Same number you can note it down. If the colony is 4 is there, you can write it down 4. So one by one you can go for the every places and you can count it out. Otherwise it became very difficult that you have already counted that part or not. It became difficult. So what you can do, use your marker to touch over there. Okay, by touching you can say that I have already counted. Other way around also, if you are not having this colony counter meter with you, what you can do, just draw some of the line on that. You may prepare the 5 square, 10 square, it is up to you. If the more than 100 colonies are there, you need to prepare more a square you need to prepare. If it is the lesser, then you may go for it by yourself. So it is discretion of any researcher, how you may go in. Other than that, when you are having the advanced kind of a colony counter meter are coming, what is there? There you have already fixed your plate. Plate is fixed, and when you start to touch, just touching your knobs onto that plate, it is going to consider that I'm looking to take it as a colony. It is already coupled with our laptop. Okay, so it is having the automated counting as well, and another is the software. We need to tell that manually. That manually I'm going to consider this as a colony. This one is a colony. This one is a colony. So it is going to calculate accordingly. Okay, so colony counter meter, you can do it. Same you can do it with using your microphone. You can take the photograph one at a time. Then at the second day also you can calculate. If you are going to observe this plate on a second day, might be more than that colonies may appear, because some of the place microbes are not able to grow within 24 hours. It may take 48 hours. Okay, so on a second day, it is on after 24 hours, we need to take the photographs. Either you may take the photographs or you may calculate by itself, by yourself. It is up to you. So every person having their own way to do the experimentations. So in a microbiology, generally we are going to have at least additional day to get observe your results. It is better that is been kept in a Thursday. So maybe on a Friday when you are having your break, at least for the record your result, it will take 20 minutes, not more than that. 15 to 20 minutes is the maximum part will be required to note it down your observations. So this colony, and let me complete. Yeah, someone has asked about the vertex, right? Yes. Vertex is nothing but a platform. It is continuously rotating or moving. So anything you are going to keep over there, something is rotating. So it will start to make a disturbance in a liquid. To make a disturbance, there is a basic thing is what? Sometimes you are having your, just 1 second. It is like anything pen or something, tubes are there. What you need to do? A tapping. Append of tubes will come. Any sort of the tubes will be there. When it is called the gentle tapping, you must be careful. And just check it out which finger I'm using. How I have held the tubes. That is the essential part to be get understand. Okay, you can do it. It is not advisable. Okay, you can do anything. You can agitate. You are looking to agitate it. Why it is not required, or what happened if the glass tube is or plastic is there, and it may happen that it may touch to your cotton plug, or may your caps get fall down somewhere. So gentle tapping is required. If the tube is there, we have to hold at least at the 2/3 portion of it. That is 33% is upper side, 66% is the below. That is the optimum and advisable place to hold the tubes. Whether it's a smaller tube, larger, append of tube, centrifuge tube, glass tube, sugar tube, while tapping. And tapping is required to do. Those who are having the larger nails, be careful. sometimes the glass tubes are get broken down. They are going to disturb themselves itself and other one, because when a tube is get broken down, you are tapping your nearby friends is always there. You are going to make a harm to other also. So be careful while tapping. It should not be go beyond or the near to the eyes of any person. By chance accidentally may happens, then you and your friends are in problem, in a trouble. Okay, so it is some know-hows how to process in a lab. It should not be done at the very lower stage. You are not able to manage. It should be at the certain level near to your face. So what is going on, you can observe. Okay, it should not be like that. Okay, we are doing. If you are doing, work can be done, but it may get lost. Okay, so these are the some basic knobs. So vertex is nothing but this is the place, it is continuously rotating. Platform is rotating, so you can keep it over it. Here you can increase and decrease their speed. So which kind of a solutions you are having, accordingly you have to manage their speed. The RPM, rotation per minute, RPM is always there. If you are having your glass tube, sugar tube, and it is you are running at RPM of 2000, just keeping and your tubes is going to get thrown away. Okay, so what you can do, first, if you are going to use the vertex, you need to keep over there, then manually, gently increase your speed. So you need to hold it. Tube should be get held because platform is rotating. If you are not firm in your holding, then while rotations, tubes will get fall away at another place. So currently we are not going to use this kind of a vertex right now. Just a basic gentle tapping is our first target. And when you are tapping, not at the higher level, okay, near to their butt, okay, at the below the region, at the butt you are going to just tap. Okay, so it is much important to understand what you are going to do in the next day. Okay, so when you are going today, you are going to get some tube, same. It is remain in a room temperature. So we are going to just observe whether it is get contaminated or not. If contamination is there, that means some flows are there. Either your cotton plugs were not proper. So within a week, microbes has gone inside. If it is also there, then a problem. Okay, and just check it out. At the below bottom, just tap it, your tubes. While tapping, you are going to observe that is there anything at the bottom area or not. So we'll easily able to observe that you have successfully prepared your nutrient broth. After 1 week period also, if there is no contamination, that means successfully first part is completed. Next is inoculation, liquid medium inoculation. So that part I'm going to say in practical session only. So I hope basic things related to spreading is clear. Fine. Same thing I'm going to demo while in practical session. Okay, now it's the next part is the pour plate technique. Okay, pour plate technique is like a pouring. I do have the plate with me. Okay, while preparing the solid media, I'm just going to pour, make it the plate solidified. And when plates are ready, I'm ready with my culture. I'm go for the either sticking or spreading. But in case of pour plate technique, what is our target? That before it gets solidified in a tubes or in a, when I am having the same thing in a tube either or in a flask is there, I'm going to inoculate it immediately at that time. Like during the spreading, what we have used our micro pipettes to take the certain amount of a liquid, right? And that we have just transferred to our Petri plates. And then after you have used your spreader, that is a glass spreader to spread over the surface. In case of a pour plate, what is the basic thing? While preparing your broth, okay, you are going to add your into agar medium directly itself. What you are going to add? You are going to certain microbes. Okay, you mix it. Okay, there also the tubes are when the solutions within the nutrient broth is there. It has been added with agar. So now it is N agar. Plus you are going to add your microbes. But after the adding the microbes, you need to close first your tube with a cotton plug and then gently mix it. Okay, you can mix in the 2 to 3 formats. Okay, mixing is required because liquid part is upper area. So it should be evenly spreaded within your about a 20 ml sugar tubes were there having the agar about a 20 ml. So what you are going to do? Additions. Okay, you are not going to mix in this way because upper side is a cotton. Okay, so you are not going to. Yes, some of the time I will say that you need to. This one is called the invert. When the tubes are there, okay, anything is there, append a tube. I'm going to say that you need to invert your tubes. Invert itself telling that all the liquid is going to touch. When I'm saying the invert, it must be done. Then only your protocol will be get succeeded. If you are going to mix in this way, it won't. In the microbiology, molecular biology experiment, DNA isolation, others are coming that you need to invert it. So we are not going to invert. We are going to just mix it. So mixing is required. You are using your palm where you are going to stuck. But be careful, it is somewhat hot in the nature. About 45 to 50 degrees centigrade is required to be. And if you are not going to complete within a time period, what is going to happen? That is what is going on. I will knock you down outside. Huh? I'm noticing since long many times. Okay. I'm tolerating, that is my limitations. Fine. I'm going to notice and the keeping in my attendance record if required. My limitations power is you are utilizing, then if going beyond threshold, I cannot resist. I'm going to say that. And you also. If you are not liking my lecture, you may keep outside. You may roam outside. You can learn online. But don't disturb. If you want to do such activity, please be seated at the last place. I will allow anyone. Okay, you may sit outside. I can keep in my record. They are not interested in my lecture. And so they are roaming outside or sitting at the last place. If I'm going to teach one by one, that is for all the lectures and all the part. I can complete the same thing within an hour. I'm investing my more energy for you people to get you understand. Okay. It is not mandatory for me to teach each point how to handle and do. I can show that this is the plates you have to do, you can complete. So don't cross the limitations in my lecture. So when you're having the plates or the tubes with you, be careful. You have added your liquid medium. Solidification is going to start at the below 40 degrees centigrade. So mixing is required to do within a given time period, and you need to pour it. Pouring is required on a plate. So if you are going to remain, you are going to say this, sir, some of the part is remain in a tube itself. Now it is of no use. It should be done within about a given time period. If you are not able to manage, your practical is get failed. So each step, the sticking is very easy. Okay, spreading is somewhat more ahead. Pouring is easy. Okay, but one tougher stage is coming. So if you are just fail to manage into one to another place, your practical is get gone. Okay, so be careful into it. So what is the use of it and how it is generally been performed? There, the pour plate techniques are being used when you are having the mixture of the bacteria or you are having the looking to understand their colony forming units. Till now, I haven't just don't discuss about the colony forming units. Means what? One bacteria is capable to produce one whole colony. Okay, so if you added the 100 microbes, it is having the capacity to generate 100 colonies. So what basic amount, if I'm going to take the 0.1 ml or the 1 ml solution, if I have taken, it should or must have at least this number of microbes to generate the colonies. So we are going to calculate in a way that how many colony forming units are there. That means how many microbes are there. So if you have started, like a solution is there, here you can see the original solution in first tube. It having here its appearance is very blurred. So it having the number of the microbes maybe a 1 lakhs. if you are going to take the 1.1 microliter out of it and you are going to pour even, you will able to find that the whole plate is full of microbes. Okay, because having the higher concentration. So what you need to do, you need to go for the serial dilutions. I hope you are knowing about the serial dilutions. Means what? You may going to take the 1.2 ml, 1 ml out of it, and you are going to add into 9 ml a liquid bronze. 1 ml is taken and the 9 is taken. Yes. 9 plus 1. So you have diluted it's up to 10 times. Once again, you take the 1 ml, you transfer to the next tube. Okay. So while transferring each step, you need to mix well. Mixing is not an invert. Okay. It is not an invert. You are going to stuck with your palm. Okay. It's going to stuck with your palm only. So each step you are going to mix and the transfer is going on. Right. Sometimes people are using the liquid as a H2 to dilute it. Sometimes people are using the nutrient broth to dilute it. So it is up to the kind of a bacteria or the place you are working. Accordingly, we need to manage. Okay, so now out of it, I may go for mixing. So if I'm going to use this kind of a solutions, maybe I have diluted up to a certain stage, you may just check it out. That's how many colonies are being observed. Okay, in the next time, when it's diluted up to 10 times again, that is like a 10 raised to minus 1, minus 2, minus 3, 10 raised to minus 4. That the dilution is done till that limit, you are going to get a certain limit of a number of the colonies. Here it is very high. Again, if you have diluted more than that, beyond your limits, if you have diluted 10 raised to minus 5 up to 10 raised to minus 10, you may prepare the plates for every. Then you will observe that after a 10 raised to minus 6 or 10 raised to minus 7 dilution, there is no any colonies observed. Okay, so what the limitations when you are looking for very isolated? What we are understanding and looking for the pure culture. That means the bacterial plates should have only same kind of a bacterial colony and in a minimum number. So it can be observed very easily. Here it is in crowded. Okay, if someone is in crowd, okay, someone has done something, it is really difficult for me to identify. Okay, but only 4 or 5 students area and they're there, and someone has spoken something, I can at least ensure that this one is the only a person has talked. Okay, it is like that. So it's isolating one to individual. So we are looking to isolate as much as less number of the colony. So for that, what we need to do? First, dilutions is required to done. Yeah, always sterile distilled water. Osmosis. That's related to osmosis you are asking, right? How much amount you have taken? And yes, when you are diluting, that is why I have told, it is depend upon the bacterial solutions. Sometimes we are using the nutrient broth, sometimes we are using the distilled water. Some bacteria can resist very easily. They're already there in a lake, right? From the water sample, it's like E. coli. Generally, we are working on E. coli. They can very easily survive into such water. Okay, same we are using over here. So they want, yeah, special kind of a bacteria are there. If acidophile is there, pyrophiles are there. If it is such so, then it's required to prepare the different nutrient medium. Yeah, otherwise osmosis problem, hyper or hypotonic may arise. That's right. Okay, so this part is clear. So what is the basic methods and steps? That's what we need to do before it is not directly poured. Okay, the same amount is taken into 1 ml. 1 ml is mixed with our 20 ml hot agar. Okay, and this agar is available from where? How it has been available? You have prepared 20 ml in a sugar cube. Like nutrient broth is there today. After the autoclaving, it should not be get solidified. So we have kept in the hot condition. Okay, hot condition is there directly from the autoclave. The tubes are coming. It may be near to 100 degrees centigrade. We have to keep continuously open and there below to it, water should be kept into boiling condition. So while taking the tubes from the autoclave, you have to be careful. Yes, I may advise that you people should have at least one napkin with you. Okay, smaller or larger, you may use your handkerchiefs, not a problem, or a napkin with you, because while handling and holding, it may burn your, or it may having the some effect on your hand. While handling also, if you are coming in there, it may happen that you are not able to manage and it's get put down. Otherwise, you will throw down, because it's like a sometimes when we are going into some canteens. Currently, it having the cups of the plastics, or the cup are from the paper as coming. Earlier, it was used to with some still or the glassmates were from. So if the hot tea is given to you at a time, okay, you may hold for a while. Otherwise, you may start to change your hand. Even in the paper cup also, for a while you can hold. What could be the temperature of it? Can you say of that tea? It may be 100 degrees centigrade or less than that. First thing, temperature. What could be the around temperature? Huh? Okay, what is the less than 100? Okay, it is more between 50 to 100 or below to 50. Huh? 50 to 100. Between. What could be the temperature at the canteen whenever they are serving to you some hot tea? What could be the temperature of that tea? 80. 80 to 95. Huh? 50 above. 50 to 70. Below 200. Okay, very good, clever answer. Okay. So we haven't never thought that what we are daily doing. That is why I'm telling that those who are good in a cooking first, then those who are good in eating also important here, right? So sometimes just the cup is coming near to you, start to blow. You haven't touched even. How you come to know? Or your intellectuality or your mind is telling that this tea is not in that conditions to directly drink. Something is there. You haven't noticed, but your brain has already noticed that should I blow something. And you, if you are some little, you have start to take. Okay, then if it is higher, you will keep away. You blow it. While blowing, is it like that all the cup is get cooled down? No, only the upper area is there. Okay, so you are gradually increase and decrease your temperature. Same thing is going to while in the pour plate method at the 100 degrees centigrade shortage. So whenever it is been served, where if it is kept, your tea is kept into somewhere in jar or thermos, just think like that. It is the about 60-70 degree temperature. When it is poured into your cup, it may going to reach to between 40 to 70. And within a couple of minutes or time period, it may reach to a 40. Then it will sustain because the water having the capacity to hold their temperature for a about time period. So at the 40 to 50, it is bearable for us to drink it. Okay, so same thing is going to happen in this our case. When you are going to take the tubes, it is about 100 degree temperature. You have autoclave maybe there. Here you are going to work. You have kept your tubes on a platform on one particular stand. Now the time period has started. Ticking from 100 to 40. So what you are going to do? You are going to take your culture. You have started to manage your place. Now just take the tubes and touch to your this part on the above part of your hands. Whether it is you are able to bear it or not. Like a 40 degrees centigrade is bearable if by you, then only it get bearable by the microbes. At the hotter conditions, at 100 degrees centigrade is there. If you inoculated it, bacteria will die. Okay, immediately they are going to die. It is like that. In the hot stream, you are asked to swim. When in a swimming is done in some other place, we know that in the winter conditions, people are reluctant to go. So in a summer conditions, there will be not a problem. But you are going in like a Tulsi system and other places. Okay, there are hot streams are there. That the streams are coming out of it. So what you are going to do? You may wait that should I jump over there or not, because it's a near about 80 degrees centigrade is there. So what you will do, so you are just going to touch. The same thing is there. You have to touch first your tubes to your hands, not on a palm, because the palm has a higher capacity to tolerate the temperature. Here our skin is much sensitive on the upper hand. So you are going to touch over there. And then you realize, okay, now it's okay. Sometimes you will say, let me wait for a while. So don't wait longer. It should be at optimum temperature is coming. Then you start to pour or by using the pipettes, you are going to add your suspension. Then you will mix it. Mixing, then you are going to throw with your palm. As it is completed, you are going to, now the two things will be there. One is the plate with you and one another is a tube. So what you are going to use, your tube should be open up. Okay, tubes is open. Now you are not going to use this tube right now. So you can open their cotton plug, you can keep it aside. In this pour technique only. Okay, you are going to keep your cotton plug aside, not a problem. Then you are using this tube. Yes, while handling at a 40 degrees centigrade also, it may having the some hot pot. So what you are going to do, at the neck part, below to the neck at a 33% is gone, there you are going to hold from the beside. You are holding like this. And you are going to try to pour. Your whole hand is going to get hotter area, will be filled. So what you are going to use in these conditions. Now on the second end, plates is ready. You are going to open it. You are just going to pour. Pouring should be even. If the time is gone, then what is going to happen? That solidification is happens, but it is in layer. Okay, in angles it may happen, or sometimes solidifications may happen that if drop is done, drop by drop you are going to add that, then the solidification may happen that you will find that it's like a mountain. One is here and there, mountain like this. It should not be like that. It should be transferred in particular way. As it's completed, you can keep your tube aside or in a rack and close the plates and just rotate. So while rotation, it can be managed that if till the solidification is not completed, evenly spread it on your plate. Okay, now your task is complete. You wait for about 10 to 15 minutes because it's start to get solidified, but not completely solidified. Okay, so at least you need to wait for about 20 minutes to it get solidified. Then you need to invert it and incubate in an incubator for 24 hours. So this is the basic protocol already. So same part can be seen into our documentary here.
Pour plate method is usually the method of choice for
mobile even if you want to hurt.
counting the number of colony-forming bacteria present in a liquid specimen. Welcome to the channel Microgames Experiments. Be with us, be a laboratory expert. To perform this method, we need a biological safety cabinet, incubator, micropipette, vortex mixture, micropipette tips, and petri dish. So, let's begin with the culture media preparation. Take weight of agar media and mix with distilled water following the label on the media container. For sterilization, some media need to just boil to dissolve the media content. Some need autoclaving at 121 degrees Celsius and 15 pounds for 15 minutes. Others need to boil in microwave oven for 2 minutes. After sterilization, the media is cooled to 45 to 48 degrees Celsius in water bath. Now, we are going to main test for inoculation. Clean your workstation inside the biological safety cabinet. Bring everything needed for the pour plate technique. Label 2 sterile and blank petri dishes for 2 samples. Labeling should be done by media name, test name, method, date, etc. Take first sample and vortex it to make the homogeneous mixture.
Few things are same as before.
Shake and invert the sample well before taking
Here
it
you
for
can
testing.
do centrifuge tube.
Take 1
It is not
ml of the sample 1
having the
by
glass
a
tube. It's not
micropipette
having the
and
cotton.
pour it on the middle of the petri dish. Cover the dish immediately with its lid and discard the tips from the micropipette.
Here they are also showing the another method that into it they are going to
Do the
put
same
into
thing
the
for taking the
solution.
sample
The
2
first one is
on the
put
second
into your
petri dish.
plates. They haven't added into directly agar.
Now, bring the sterile culture media from the water bath. Pour 15 to 17 ml of the media.
Here they have done into the plates, but that part we are not going to do. Suppose that plate. Because the reason is what? The movement of that part here just is trying to do gently. It may going to touch to your plate because you are not doing that practice. How to rotate that plate is the essential part. It's not going to happen if then it's going to touch to your even the lid. So we are going to add the same thing in a tube. These all are the three major methods around the world it's been used for the isolating the journals. So as a microbiologist, these three techniques are very much essential for aerobic bacteria. While in anaerobic, I will going to tell you about it next week. How to handle the anaerobic bacteria. This all is regarding the aerobic bacteria. Get solidified. Invert the plate. That's the simple part. Fine. So this is the part regarding the four plates. So is there any first doubt? Anybody? Clear what we are going to perform? Here in documentary part they have shown that we are going to keep into directly in a tube 2 plates. But that part we are not going to do it. Just running. Yeah. In the plates they have taken, we are not going to take in a plates. What we are just understanding and going to look in a pour plate method, bacteria is going to get entrapped in all your agar. Okay, it is just another way to culture your bacteria, nothing else. You can go in a spread or in other also. Same it is done into pour plate. Here you are going to pour into it. Okay, pouring is done in two ways. What he has done that on a petri plate they have taken the bacteria and the medium is given to allow to grow. Other way, same thing you can do in a tubes. You may take some medium. You are going to add that media over it, mix over there, and then you are going to pour on a plate. So evenly, it is not going to be absorbed onto directly the surface. It is get entrapped within one part. Okay, so that is their advantage and disadvantage. That's I'm going to show that part. Yeah. So here, what's going to happen? The pour plates can be used to isolate and determine the number of cells in a population. Means if I have taken the 0.1 ml or the 1 ml solution is there, I can calculate as a colony forming units. That means what? That 0.1 ml give the 100 colonies. If I have the 1 ml, then how many organisms could be there? We can calculate it. 0.1 ml give the 100 colonies. If I have the 1 ml suspension, then how much colonies could be have? Can you calculate? 100? I haven't taken marker. What I'm telling, have you heard? 0.1, 100. 0.1 ml, it's 100. I'm telling about the 1 ml. That is saying how much? One. First is a 1,000. 1,000. Is the number is coming out? You are just not listening. That's I'm telling. Take it out your diary first and calculate. 1,000. 0.1 ml. Just listen. 0.1 ml is 100. 1 ml means how many times it is going? 10 times. So here it was 100. It's going to 1,000. Okay. In that way, we need to move ahead. If I'm going to use the word 1 liter, then 1 liter. So 1 ml is equal to 1 liter is equal to how many ml? 1 liter is equal to how many ml? 1,000. So you need to multiply your 1,000 with 1,000. So how much it will going to cover? 6 zeros. How it is pronounced? Yes, 5 plus 1, 10 lakhs. So you are going to get the 10 lakhs colony forming units. So 10 lakhs microbes were there that you can calculate. Colony forming units means what? When you started, my initial suspension solutions was just 0.1 ml. And I have received 100 colonies. Okay, if I have taken the 1 ml, then how many colonies going to attribute? 1,000. So I do have the 1 whole flask of a 1 liter. That means how many bacteria could be there? 10 lakhs. That was our target to identify or calculate our artist. So 4 plate techniques been used in this case. Spread plate techniques also been used. Yeah. 4 plate techniques and spread plate techniques. You are going to take the 0.1 ml or the 1 ml solution evenly spreaded, and how many colonies you have observed. So it is called the colony forming units. Each bacteria having the capacity to develop their colony. So number of the bacteria is how many that you are going to calculate. Okay, so here we can see that 4 plate can be used to isolate as well as determine the number of cells in a population. Getting understood? You have taken the 0.1 ml, 100 colonies. If you have taken the 1 ml, you are going to get the thousands. So your 0.1 ml suspension having the about 100 numbers of bacteria. So when you are working, what we need to do? We need to, in next experimentations, if I want to go ahead and I want less than 100 colonies, then I should dilute again 10 times. Okay. I may go for the 10 times or 5 times. That is up to our limitations. You can go for the 2-time dilutions. Okay, that is a practice is required. That 100, I just want a 10 colonies. So I will go for the 10-time dilution initially, and then I'm going to take that about a 1 ml from it and pouring in this experimentation. So number of the bacteria within my 1 liter flask is how many that need to calculate. So that is the main advantage of this both the technique is there. Plate containing between 30 to 300 colonies are counted. Maximum capacity for us is about 300 colonies. Okay, if more than that is there, you are not able to calculate very easily because it is whole spaces get occupied. Then you are going to say that it's a lawn growth. As you are roaming in a lawn, green grass is there, right? Lawn is there. You are going to return that I have done the experiment and I observed lawn growth. That means number of colonies really difficult to calculate. Then the total number of colonies equal to the number of viable microbes in the sample that are capable of growing into medium that used. That means how many number of colonies are there? Same number of the microbes may be present into my 0.1 or the 1 ml culture, what I have taken. Fine. Then the next disadvantage, that's the major essential part. Temperature of the soft agar is 45 degrees centigrade. Okay, here what we are going to use, if below that, if you are going to, temperature is reduced down, and then you are going to pour or add your sample into it, then it start to get solidified within the tube. Then the experiment will not going to run. So one limitation is it, and another are the sensitive bacteria. Even at a 45 degrees centigrade, they may die. So we cannot work out such sensitive bacteria to be get processed over here. Okay, both the surface as well as submerged colonies are going to form. That submerged colonies may be small and hence can be missed during the enumeration. Yeah. When you are going to do in a spread plate techniques and counter, okay, colony counter, it's easy to observable. But when it is submerged within it, it's difficult to identify because you have to use the colonies. Okay, light you are using. Sometimes it is upper and lower part is there. You may miss into calculations. So it's become their disadvantage. Also, submerged colonies are difficult to pick up for the further inoculation. If you are looking or interested that I want to do certain work on that, okay, then it's much difficult because the colonies is being formed within the agar. Okay, it is like some ice cube is there. Within the ice cube, some portion is been get entrapped. How to take that part? So you can may, if you are interested, then use the earlier I have shown the nichrome wire. Okay, without look, it is like a pin is there. Okay, pinpointed. Such nichrome wire we have to use just to poke over till the colony is going to reach. You may take and you may transfer. So in pour plate techniques, we can use such kind of a just a pinpointed nichrome wire. Okay, so it's somewhat limitations, but can be managed in that way. So I hope this clear up to till. So this can be, 4 techniques can be asked into whole 8 marks till now. Okay, whole techniques, maybe any questions like a advantage or disadvantage can be asked into 2 marks, 4 marks, anything can be happen. Okay, same questions can be asked into 4 marks. In essential, you have to draw it very well. Till now, you haven't drawn anything, right? Just if I'm looking to, if it is there, okay, the spread part is there. Such you, if you are not doing the draw the hands, it's okay. But lookful part you have to do. The beaker is required to prepare. Glass rod, you need to draw it. Burner, if you are not going to draw proper the same burner, it's okay. Okay, but it is the some burner like this and fumes are coming. So you need to write it's the burner. Otherwise, I am not able to understand. Labeling is required to be done properly. So in each of part, in microbiology, especially in such papers, are required to have the essential figure component. Labeled figure components should be drawn properly, and then you need to write their procedure. What are the steps are required? If it is of a higher weightage marks, you need to write it about their advantage and disadvantage also. Otherwise, you say that just I have written only the part. If it is 8 marks or the 6 marks, you have to give the whole package. Okay, if it is just 1 or 2 marks, you can write it down accordingly. The time limitations is also there. You have to set the priority while writing.
This.
Okay, these are the advantage until the disadvantage into.
Pour
Oh, this was the just last point into it. So I hope first whole 3 experimentations now onwards we are going to do. That is why I have taken longer time. In practical part, when we are going to do, I'm just going to demo some of the portions. I will take brief some theory. Okay, so if I'm going to keep on the deliver on the theory in a lab, we are not going to complete before 2 o'clock. So on the next, every time we are going in a practical sessions, briefly I'm going to say about what we are going to do in a lab. Okay, into it, and then we will move. So today I will come first in a class. Within a 10 or 15 minutes, we are going to move in a lab. What we are going to do in a lab, that I will tell you when I will come. Okay, till this, any having any questions into this 3 basic techniques? These are the very much essential techniques that is why I have given more time. This is the core microbiological techniques. Whenever in a world you are going, if you are a microbiologist, you need to understand and learn these 3 things. Continuously, same thing is going to. Fine. So just I'm ending here. You can put it down your attendance, those have left into given link. Okay, so see you directly in a next. There, I think we are not going to, those who are in online mode right now also, they are not able to perform it. They can use their YouTube videos or they can surf on the online. Those who are in offline mode, for them, I am able to take in a lab. Okay, that is my limitations. I cannot take my lab to their own home.